Hello and welcome to Nighttime Science. Tonight we journey into the quiet world of genetics and DNA, where the instructions for all life rest in molecules so small they cannot be seen, yet so powerful they shape every living thing on Earth. In the year 1869, a young Swiss physician named Friedrich Miescher made a discovery that would eventually change our understanding of life itself. Working in a laboratory in Tübingen, Germany, Miescher was studying the chemical composition of white blood cells, which he collected from the pus-soaked bandages of hospital patients. As he worked with these cells, extracting and analyzing their components, he isolated a substance from the cell nuclei that behaved unlike any protein or fat known to science at the time. This substance was rich in phosphorus, slightly acidic and remarkably stable. Miescher called it nuclein because it came from the nucleus. What he had discovered, though he could not have known it then, was deoxyribonucleic acid, DNA. For decades after Miescher's discovery, scientists continued to study nuclein, slowly unraveling its properties. By the early 20th century, researchers had determined that this mysterious substance was made of smaller units called nucleotides. Each nucleotide consisted of three parts: a sugar molecule, a phosphate group, and one of four nitrogen-containing bases. These bases were given the names adenine, thymine, guanine, and cytosine. Yet even as the chemical composition became clearer, the true purpose and structure of DNA remained hidden in shadow. Throughout the 1930s and 40s, evidence slowly accumulated that DNA, not protein, was the carrier of genetic information. Experiments with bacteria showed that DNA could transfer traits from one organism to another. But understanding what DNA did was different from understanding how it worked. The structure of DNA, the precise arrangement of its atoms in three-dimensional space, held the key to its function. Without knowing the structure, scientists could not explain how DNA stored information, how it copied itself, or how it passed traits from parent to offspring. The race to discover DNA's structure intensified in the early 1950s. At King's College London, a scientist named Rosalind Franklin was using a technique called X-ray crystallography to examine DNA fibers. When X-rays pass through a crystallized substance, they scatter in patterns that reveal the arrangement of atoms within. Franklin was exceptionally skilled at this painstaking work. She produced X-ray images of DNA that were remarkably clear, showing a distinctive cross pattern that suggested a helical structure. Her photographs, particularly one known as Photo 51, contained crucial clues about DNA's architecture. Meanwhile, at Cambridge University, two scientists were building physical models to try to deduce DNA's structure. James Watson, a young American biologist, and Francis Crick, a British physicist turned biologist, were combining all available evidence, chemical data, X-ray images, and the known rules of chemistry to solve the puzzle. In early 1953, Watson saw Franklin's Photo 51, though through circumstances that remain controversial to this day. The image revealed what he and Crick needed to know. On February 28, 1953, Watson and Crick completed their model of DNA's structure. They had built it from metal plates and rods in their Cambridge laboratory, a three-dimensional sculpture representing molecules too small to see. What they had constructed was a double helix, two long strands twisted around each other like a spiral staircase. The sugar and phosphate groups formed the outer rails of this staircase, while the bases pointed inward, forming the steps, each base on one strand paired with a specific base on the other strand. Adenine always paired with thymine, and guanine always paired with cytosine. These pairing rules meant that the two strands were complementary. If you knew the sequence of bases on one strand, you automatically knew the sequence on the other. The elegance of this structure was breathtaking. It immediately suggested how DNA could copy itself. The two strands could separate, and each could serve as a template for building a new partner strand. It showed how information could be stored in the sequence of bases along the strand, like letters in an alphabet spelling out instructions. And it revealed how that information could be both stable and changeable, protected yet accessible. Watson and Crick published their discovery in the journal Nature on April 25, 1953. Their paper was remarkably brief, just over 900 words. Near the end, they included what may be one of the greatest understatements in scientific literature. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. With characteristic British restraint, they were announcing that they had found the secret of life itself. Rosalind Franklin's contribution to this discovery was immense, though she did not share in the credit during her lifetime. She died of ovarian cancer in 1958 at the age of 37, possibly due to her extensive exposure to X-rays during her research. The Nobel Prize for the discovery of DNA's structure was awarded in 1962 to Watson Crick and Maurice Wilkins, who had worked with Franklin at King's College. The Nobel Prize is not awarded posthumously, and so Franklin's name was absent from the ceremony. In the decades since, historians of science have worked to restore her rightful place in the story, recognizing that her experimental work provided essential evidence for one of biology's greatest breakthroughs. The molecule they had discovered, this double helix of DNA, exists in nearly every cell of your body as you listen to these words. It is vanishingly small, yet contains vast amounts of information. The DNA in a single human cell, if stretched out straight, would measure about 2 meters long. Yet it fits inside a nucleus only about 6 micrometers in diameter, roughly 1/50th the width of a human hair. This is possible because DNA is extremely thin, only about 2 nanometers wide. To put this in perspective, if you magnified a DNA molecule until its width matched that of a piece of string, then at the same scale, a human hair would be as wide as a football field. Every cell in your body, with a few exceptions like red blood cells, which have no nucleus, contains the complete instruction manual for building and maintaining you. This manual is divided into 46 volumes called chromosomes, arranged in 23 pairs. One chromosome in each pair came from your mother, carried in the egg cell that would become you. The other came from your father, carried in the sperm cell that fertilized that egg. These 46 chromosomes together constitute your genome, your complete genetic inheritance. As you rest and let these thoughts settle, consider that inside you right now, at this very moment, your DNA is being read, copied, and translated by molecular machines smaller than bacteria. Instructions written in an alphabet of just 4 letters, A, T, G, and C, are directing the construction of thousands of different proteins, each one folded into a precise 3-dimensional shape that allows it to perform its specific task. Your heart beats, your lungs draw breath, your neurons fire, all because of information encoded in the sequence of those 4 bases, wound into a double helix, coiled and folded and packed into the nucleus of each cell. From Miescher's nucleon to Watson and Crick's double helix, the journey to understand DNA took nearly a century. The journey to understand what DNA does and how it does it would take many more decades and is continuing still. In the sections ahead, we will explore the architecture of genetic material in greater detail, learning how DNA is packaged and organized. We will follow the process by which DNA copies itself with extraordinary accuracy. We will discover how the information in DNA is translated into the proteins that build and run your body. And we will examine the ways DNA can change, repair itself, and respond to the environment, creating the diversity of life we see all around us. For now, as this first part of our journey comes to a close, let the image of the double helix rest gently in your mind. Two strands wound around each other, complementary and elegant, containing in their sequence the instructions for life itself. The story of DNA is the story of you, written in molecules, spelling out in four-letter words everything you are and everything you might become. If you're finding this journey through the quiet science of genetics calming and informative, consider subscribing to Nighttime Science for more gentle explorations of the natural world and the mechanisms of life. To truly understand DNA, we must look more closely at its architecture, the precise arrangement of atoms and molecules that allows it to store, protect, and transmit the information of life. The double helix that Watson and Crick discovered is often compared to a twisted ladder, and this comparison captures the essential geometry. The two sides of the ladder are made of alternating sugar and phosphate groups, linked together in a chain that runs the entire length of the DNA molecule. The sugar in DNA is called deoxyribose, a 5-carbon ring that gives the molecule part of its name. Each deoxyribose sugar connects to a phosphate group, which in turn connects to the next sugar, forming what biochemists call the sugar-phosphate backbone. The rungs of this molecular ladder are the base pairs. Projecting inward from each sugar molecule is one of the 4 nitrogenous bases: adenine, thymine, guanine, or cytosine. These bases are flat ring-shaped molecules that stack on top of one another like plates in the center of the helix. The specific pairing rules that Watson and Crick discovered, adenine with thymine, guanine with cytosine, are not arbitrary. They arise from the geometry and chemistry of the bases themselves. Adenine and guanine are larger molecules called purines, each composed of 2 connected rings of atoms. Thymine and cytosine are smaller molecules called pyrimidines, made of single rings. When a purine pairs with a pyrimidine, adenine with thymine or guanine with cytosine, the resulting pair has exactly the same width. This uniform width allows the double helix to maintain a consistent diameter along its entire length. If 2 purines tried to pair, they would be too wide. If 2 pyrimidines paired, they would be too narrow. The structure itself enforces the pairing rules. These base pairs are held together not by strong chemical bonds, but by weaker interactions called hydrogen bonds. Adenine and thymine form 2 hydrogen bonds with each other, while guanine and cytosine form 3. These hydrogen bonds are strong enough to hold the 2 strands together, but weak enough to be separated when necessary. For instance, when DNA needs to be copied or when its information needs to be read. The double helix is thus both stable and accessible, a molecular structure that protects information while allowing it to be used. The 2 strands of the double helix run in opposite directions, a feature called anti-parallel orientation. Each sugar-phosphate backbone has a directionality, with one end called the 5' end and the other called the 3' end, named after the carbon atoms in the sugar molecule. In the double helix, if one strand runs from 5' to 3' in one direction, the complementary strand runs from 5' to 3' in the opposite direction. This anti-parallel arrangement has important consequences for how DNA is copied and how its information is read. The double helix completes one full twist every 10 base pairs or so. Along the length of the molecule, this twisting creates two grooves of unequal width, a major groove and a minor groove. These grooves are important because they allow proteins to interact with DNA, reading the sequence of bases without having to separate the two strands. Proteins can nestle into these grooves and recognize specific sequences of bases, binding to DNA at precise locations to turn genes on or off, or to begin the process of copying DNA. But DNA does not exist as a naked double helix floating freely in the cell. If it did, it would be impossibly tangled and vulnerable to damage. Instead, DNA in human cells is packaged in an elaborate hierarchy of structures that compact it, organize it, and protect it. The first level of packaging involves proteins called histones. Histones are small, positively charged proteins that DNA winds around like thread on a spool. 8 histone proteins group together to form a structure called a histone octamer. DNA wraps around this octamer 1 3/4 times, making about 147 base pairs of contact with the histone surface. This combination of DNA wrapped around histones is called a nucleosome. Nucleosomes are the fundamental repeating unit of chromosome structure. If you could see DNA under an extremely powerful microscope, it would look like a string of beads, each bead a nucleosome, with short stretches of DNA connecting one nucleosome to the next. This beads on a string structure represents the first level of DNA compaction, reducing the length of the DNA fiber by about 7-fold. But even this is not compact enough to fit all the DNA into a cell nucleus. The string of nucleosomes further coils into a thicker fiber, and this fiber loops and folds into increasingly complex structures. Additional proteins help organize these loops and anchor them to a protein scaffold that forms the interior framework of the chromosome. Through these progressive levels of folding and coiling, DNA that would stretch for 2 meters if laid straight is compressed into chromosomes that collectively occupy a nucleus only a few micrometers across. This packaging is not merely structural. It also plays a crucial role in controlling which genes are active and which are silent. DNA that is tightly wrapped around histones and compacted into dense structures is generally inaccessible to the proteins that read genes and make RNA copies. This tightly packed form of DNA is called heterochromatin, and genes within heterochromatin are usually turned off. DNA that is more loosely packed is called euchromatin, and this form is accessible for genes to be read and expressed. The cell can modify histones by adding small chemical groups to them, a process we will explore more fully when we discuss epigenetics. These modifications can make histones grip DNA more tightly or more loosely, effectively turning genes on or off without changing the DNA sequence itself. The architecture of genetic material is thus dynamic, responding to the cell's needs by making certain regions of DNA accessible while keeping others closed. Each of the 46 chromosomes in a human cell is a single, enormously long DNA molecule packaged with histones and other proteins. The largest chromosome, chromosome 1, contains about 249 million base pairs. The smallest, chromosome 21, contains about 47 million base pairs. Together, all 46 chromosomes contain approximately 3.1 billion base pairs. If you were to print out the human genome as text, using the letters A, T, G, and C to represent each base, the printout would fill about 200 telephone books, each 1,000 pages long. At the ends of each chromosome are special structures called telomeres, which we will examine in detail later. For now, it is enough to know that telomeres act as protective caps, like the plastic tips on shoelaces that prevent them from fraying. With each cell division, these telomeres shorten slightly, functioning as a kind of molecular clock that counts down the number of times a cell can divide. Chromosomes also have a pinched region called the centromere, where proteins attach during cell division to pull the chromosomes apart into daughter cells. The position of the centromere varies from one chromosome to another. Some chromosomes have the centromere near the middle, creating two arms of roughly equal length, while others have it offset toward one end, creating one long arm and one short arm. Most of the time, chromosomes exist in a relatively loosened state, spread throughout the nucleus. In this form, different regions can be accessed by the cellular machinery that reads genes and copies DNA. But when a cell prepares to divide, the chromosomes condense into their most compact form, becoming short, thick structures that can be seen under a light microscope. This condensation protects the chromosomes during the complex process of cell division, ensuring that each daughter cell receives a complete set of genetic instructions. The architecture we have described, base pairs held together by hydrogen bonds, strands running in opposite directions, DNA wrapped around histones, nucleosomes coiled into fibers, fibers folded into chromosomes, is the same in nearly all human cells. Whether the cell is a neuron firing in your brain, a muscle cell contracting in your heart, or an immune cell patrolling your bloodstream, it contains the same DNA organized in the same way. What makes these cells different from one another is not the DNA itself, but which parts of the DNA are packaged loosely enough to be read and which are packed away in silence. As you lie here listening, your cells are constantly adjusting this architecture, loosening some regions to allow genes to be expressed, tightening others to keep genes quiet. This is part of what makes you a dynamic living being rather than a static collection of molecules. Your genome is not a fixed instruction manual, but a responsive document, adjusting its availability to match your cell's changing needs. In the soft darkness of the nucleus, invisible to any microscope except the most powerful, the double helix winds around histone spools, coils into chromatin fibers and folds into chromosomes. This elaborate architecture allows 6 billion base pairs, 3 billion from your mother, 3 billion from your father, to fit into a space so small that millions of nuclei could fit in a single drop of water. And yet, despite this incredible compaction, the cell can still access any gene it needs at any time to respond to the demands of life. The complete set of genetic instructions contained in your 46 chromosomes is called the human genome. This genome is vast, containing approximately 3.1 billion base pairs of DNA. To put this number in perspective, if you could read one base pair per second, working 24 hours a day without stopping, it would take you nearly 100 years to read your entire genome from beginning to end. For most of human history, the contents of this genetic library remained entirely unknown. Scientists knew that genes existed, that they were made of DNA, and that they carried instructions for building proteins. But which stretches of DNA were genes? How many genes there were and what most of those genes did remained mysteries waiting to be solved. In the year 1990, an ambitious international collaboration began with the goal of reading or sequencing the entire human genome. This effort, called the Human Genome Project, brought together scientists from 20 countries. The task before them was immense. The technology for reading DNA sequences existed, but it was slow and expensive. Sequencing the 3 billion base pairs of the human genome would require advances in technology, computer analysis, and international coordination on a scale never before attempted in biology. The project proceeded in stages. Scientists first created maps of the chromosomes, identifying landmarks and markers that would help them keep track of which pieces of DNA belonged where. They developed faster methods for sequencing DNA, moving from reading individual letters one at a time to reading thousands simultaneously. They built powerful computers and wrote sophisticated software to assemble millions of short DNA sequences into longer stretches, eventually reconstructing entire chromosomes. In June of 2000, scientists announced that they had completed a working draft of the human genome. Standing in the East Room of the White House, President Bill Clinton declared that humankind was learning the language in which God created life. The achievement was remarkable. A map of human genetic instructions that had taken 10 years, $3 billion, and the coordinated effort of thousands of scientists to produce. But the draft was incomplete. Certain regions of the genome had proven too difficult to sequence with the technology available at the time. These regions contained long stretches of highly repetitive DNA sequences that confused the sequencing machines and the computer programs trying to assemble the fragments. About 8% of the genome remained unread, a series of gaps in an otherwise complete map. For more than 2 decades, those gaps remained. Scientists continued to study the 92% of the genome that had been sequenced, learning which regions contain genes, which were involved in controlling those genes and which seemed to have no function at all. But the missing 8% nagged at researchers who wanted to complete what they had started. Finally, in March of 2022, a team of scientists announced that they had filled in the gaps. Using newer sequencing technologies that could read much longer stretches of DNA at once, they had successfully sequenced the remaining 8% of the human genome. After more than 30 years, the human genome was at last complete. Every chromosome, every gap filled, every base pair accounted for. What did this complete genome reveal? The first surprise was the number of genes. Before the Human Genome Project, scientists had estimated that humans might have as many as 100,000 genes. This seemed reasonable. Humans are complex organisms, and complexity should require many genes. But as the genome was sequenced and analyzed, the number kept dropping. The final count was startling. Humans have only about 20,000 to 25,000 protein-coding genes. This is not many more than a tiny roundworm called C. elegans, which has about 20,000 genes despite having only about 1,000 cells in its entire body. It is fewer genes than many plants have. Rice, for instance, has more than 30,000 genes. Clearly, the complexity of an organism is not simply a matter of counting genes. What matters is how those genes are regulated, how their products interact with one another, and how their expression is coordinated in time and space. The 20,000 or so genes in the human genome account for only about 1.2% of the total DNA sequence. If the genome were a book, the genes would be like individual sentences scattered through an enormous volume, separated by long stretches of text that do not code for proteins. For many years, scientists referred to this non-coding DNA as junk DNA, assuming it had no function. This assumption is proven to be largely incorrect. While some non-coding DNA may indeed be junk, remnants of ancient viral infections or evolutionary relics with no current purpose, much of it plays important roles in regulating genes. Non-coding regions contain sequences that act as switches, turning genes on or off in response to signals. They contain sequences that determine how much protein is made from a gene. They contain sequences that help organize the three-dimensional structure of chromosomes inside the nucleus, bringing distant genes close together or keeping them far apart. Some non-coding DNA is transcribed into RNA molecules that do not get translated into proteins but instead perform regulatory functions themselves. These regulatory RNA molecules can control which genes are expressed, how much protein is made, and even which parts of a gene's RNA are included in the final message. The genome is not just a list of protein recipes, but a complex regulatory document full of instructions about when, where, and how much of each recipe to use. About 45% of the human genome consists of repetitive sequences, stretches of DNA where the same pattern repeats over and over. Some of these repetitive sequences are transposons, sometimes called jumping genes, which are DNA sequences that can copy themselves and insert the copies in new locations in the genome. Most human transposons are no longer active. They have accumulated mutations over millions of years that prevent them from jumping. But they remain in the genome, molecular fossils of an ancient arms race between our ancestors' DNA and these self-replicating genetic elements. Other repetitive sequences are tandem repeats, where a short sequence of DNA is repeated many times in a row. These repeats are often found near the centromeres of chromosomes or at the telomeres at the chromosome ends. The number of repeats can vary from person to person, contributing to genetic diversity without affecting genes themselves. Some tandem repeats are used in DNA fingerprinting and paternity testing, precisely because they vary so much between individuals. The human genome also contains about 20,000 pseudogenes, sequences that look like genes but cannot produce functional proteins. Most pseudogenes are the remnants of once functional genes that have accumulated mutations over evolutionary time, disabling them. Some pseudogenes were created when a working gene was accidentally duplicated, and one copy then accumulated mutations because there was no selective pressure to keep both copies functional. Pseudogenes are like old, broken machines in a factory warehouse, still present but no longer in service. Scattered throughout the genome are sequences that originated from viruses. At some point in our evolutionary past, retroviruses infected our ancestors' cells, inserted their genetic material into the genome, and then were passed down through generations. These viral sequences now make up about 8% of the human genome. Most are inactive and harmless, but some have been co-opted for useful purposes. For example, a gene essential for placenta formation in mammals originally came from a retrovirus. As you rest here in the darkness, perhaps you can imagine your genome as a vast library. The shelves stretch in all directions, holding 3.1 billion volumes arranged in 46 sections. Most of the volumes are not books at all, but regulatory documents, notes, catalogs, and ancient texts in languages no longer spoken. The actual instruction manuals, the protein coding genes, are scattered throughout this vast space, occupying only a small fraction of the shelves. Yet everything is interconnected. The notes explain when to read certain instruction manuals. The catalogs index where different manuals are located. Even some of the ancient texts, though no longer readable, serve as spacers and markers that help organize the collection. This library is not static. Each time a cell divides, the entire library must be copied. Each time a cell needs to make a protein, specific instruction manuals must be located, retrieved, and read. The organization of the library, which sections are open to readers and which are closed, which volumes are on prominent display and which are stored in locked archives, changes constantly in response to the cell's needs. Your genome is both shared and unique. If we were to compare your genome to that of another person chosen at random, we would find that you are about 99.9% identical. Out of 3 billion base pairs, you differ at only about 3 to 4 million positions. This might sound like a lot, but it represents just 0.1% of your total genome. These small differences, called single nucleotide polymorphisms or SNPs, contribute to the variation we see between people. Differences in height, eye color, susceptibility to diseases, and countless other traits. Some regions of the genome are more variable than others. Genes involved in immunity, for instance, show high levels of variation because different variants may protect against different diseases. Genes involved in essential cellular processes, by contrast, show very little variation because any change tends to be harmful. The genome is a patchwork of conserved and variable regions reflecting the balance between the need for stability in essential functions and the benefits of diversity in responding to a changing environment. The completion of the human genome sequence was a milestone, but in many ways it marked the beginning rather than the end of a journey. Knowing the sequence is like having a book in a foreign language. You can see the words, but understanding what they mean requires additional work. Scientists are still mapping which sequences are genes, which regulatory elements control those genes, how genes interact with one another, and how genetic variation contributes to traits and diseases. The genome is a reference point, a foundation upon which decades of future research will build. As you lie here in the quiet, your genome rests in the nucleus of each cell, a vast instruction set written in a 4-letter alphabet. It is simultaneously ancient and modern. Ancient in that much of it has been passed down through millions of generations. Modern in that unique combinations arose when your parents' genomes merged to create you. It is simultaneously shared and unique. Shared in that it makes you unmistakably human. Unique in the specific variations that make you unlike any other person who has ever lived or ever will live. Every time a cell divides, it must create a complete and accurate copy of its entire genome to pass on to each daughter cell. This process, called DNA replication, is one of the most fundamental and remarkable events in biology. The precision required is extraordinary. Out of the 3 billion base pairs in the human genome, replication introduces an error only about once in every 10 billion base pairs copied. To put this in perspective, if you were to copy the entire Encyclopedia Britannica by hand 10 times over, making the equivalent error rate, you would make only a single mistake. The elegance of DNA replication was apparent from the moment Watson and Crick discovered the structure of DNA. The complementary base pairing, adenine always with thymine, guanine always with cytosine, immediately suggested a copying mechanism. If the 2 strands of the double helix were separated, each strand could serve as a template for building a new partner strand. Where there was an adenine on the template strand, a thymine would be added to the new strand. Where there was a guanine, a cytosine would be added. Following these simple rules, each old strand would direct the synthesis of a new strand, resulting in 2 complete double helices where there had been one. This mechanism is called semi-conservative replication, because each new double helix conserves one strand from the original molecule and synthesizes one new strand. After replication, you have 2 DNA molecules, each consisting of one old strand and one new strand. This was not the only copying mechanism scientists could imagine. They also considered the possibility that the two strands might remain together and somehow serve as a template for building an entirely new double helix, or that the two strands might break into fragments that would then serve as templates. But in 1958, scientists Matthew Meselson and Franklin Stahl performed an elegant experiment that proved replication is indeed semi-conservative. Replication begins at specific locations on the chromosome called origins of replication. The human genome has thousands of these origins spaced at intervals along each chromosome. At each origin, specialized proteins recognize the DNA sequence and begin the process of opening up the double helix. The two strands are separated, forming a structure called a replication bubble. As the strands separate, they expose the bases, making them available to serve as templates. The enzyme responsible for actually copying DNA is called DNA polymerase. This molecular machine moves along the separated strand, reading the template and adding complementary nucleotides to build the new strand. DNA polymerase is remarkably accurate, but this accuracy comes with a constraint. It can only add nucleotides to the 3' end of a growing strand. It cannot start building a strand from scratch. It needs a short starting piece called a primer to which it can add nucleotides. These primers are made by a different enzyme that synthesizes short stretches of RNA, a molecule chemically similar to DNA but with some important differences. Once the RNA primer is in place, DNA polymerase can begin adding DNA nucleotides to extend it. Later, other enzymes will remove the RNA primers and fill in the gaps with DNA, creating a continuous strand. The requirement that DNA polymerase can only extend a strand in one direction creates an interesting problem. Remember that the two strands of DNA run in opposite directions. They are anti-parallel. At a replication fork, where the two strands are being separated, one template strand runs in the 3' to 5' direction, while the other runs in the 5' to 3' direction. DNA polymerase can move smoothly along the template strand that runs 3' to 5', synthesizing a new strand continuously in the 5' to 3' direction. This continuously synthesized strand is called the leading strand. But what about the other template strand? DNA polymerase moving in the same direction as the replication fork on this strand would be trying to synthesize in the wrong direction, 3' to 5', which it cannot do. The solution is ingenious. This strand, called the lagging strand, is synthesized in short fragments. As the replication fork opens up, a short stretch of template becomes available. An RNA primer is laid down, and DNA polymerase synthesizes a short segment of DNA, perhaps 1 to 2,000 nucleotides long. Then, as more template becomes available, another primer is laid down and another fragment is synthesized. These short fragments, called Okazaki fragments after the scientist who discovered them, are later joined together by an enzyme called DNA ligase to form a continuous strand. The accuracy of DNA replication depends on multiple layers of error prevention and correction. The first layer is simply the chemistry of base pairing. Adenine fits well with thymine, and guanine fits well with cytosine, while mismatches do not fit as snugly. DNA polymerase is shaped to favor correctly paired bases, making it more likely to incorporate the right nucleotide. But even with this preference, mistakes happen. DNA polymerase occasionally adds the wrong nucleotide, perhaps a guanine opposite a thymine instead of the correct adenine. When this occurs, the second layer of accuracy comes into play: proofreading. DNA polymerase has a built-in error detection mechanism. If a wrong nucleotide is added, the enzyme pauses. a special region of the polymerase called the exonuclease domain, then removes the incorrectly added nucleotide. The enzyme backs up one step, removes the error, and then tries again. This proofreading activity reduces the error rate by about 100-fold. Even after DNA polymerase has finished its work and moved on, a third layer of quality control remains. A system called mismatch repair scans the newly synthesized DNA looking for base pairs that do not match correctly. When a mismatch is found, the repair system must determine which strand contains the error: the newly synthesized strand or the template strand. In bacteria, this is accomplished by checking for chemical modifications that distinguish old DNA from new. The repair system cuts out a segment of the new strand containing the error and fills in the gap with the correct sequence. This mismatch repair system reduces the error rate by another 100 to 1,000-fold. Through these layered mechanisms, accurate base pairing, proofreading by DNA polymerase, and mismatch repair, the overall error rate drops to approximately 1 mistake per 10 billion base pairs. Given that the human genome contains 3 billion base pairs, this means that when a cell divides, the new copies contain on average less than 1 error across the entire genome. This extraordinary fidelity is essential for maintaining the integrity of genetic information across generations. DNA replication requires more than just DNA polymerase. Many other proteins participate in the process, each playing a specialized role. Helicase enzymes unwind the double helix, breaking the hydrogen bonds between base pairs to separate the strands. Single-strand binding proteins coat the separated strands, keeping them from reforming base pairs with each other. Topoisomerase enzymes relieve the tension created by unwinding the helix. As the two strands separate, the DNA ahead of the replication fork becomes overwound, and topoisomerase cuts and rejoins the DNA to relax this tension. Primase synthesizes the RNA primers that DNA polymerase needs to begin synthesis. DNA ligase seals the gaps between Okazaki fragments on the lagging strand. The entire replication process is coordinated with remarkable precision. Multiple replication forks move along the chromosome simultaneously, each one proceeding at a rate of about 50 nucleotides per second in human cells. Despite this speed, the process is carefully controlled. Checkpoint mechanisms ensure that replication is complete before the cell proceeds to divide. If problems arise, if DNA is damaged, if replication stalls, if errors accumulate, the cell can pause, giving repair systems time to fix the problems before continuing. In human cells, replicating the entire genome takes several hours. During this time, approximately 6 billion nucleotides must be added, 3 billion for each of the two new strands. The nucleotides themselves must be synthesized by the cell, requiring energy and raw materials. Each new nucleotide added requires breaking a high-energy bond, releasing energy that drives the synthesis forward. Replication is thus not only accurate but energetically expensive, consuming a significant portion of the cell's resources. As you rest here, perhaps you can imagine the quiet industry taking place in dividing cells throughout your body. In the bone marrow, blood cell precursors are dividing, each one copying its genome before splitting into two daughter cells. In the lining of your intestines, epithelial cells are dividing to replace those lost to the constant wear of digestion. In a healing wound, skin cells are dividing to close the gap. In each of these cells, DNA polymerase molecules march along separated strands, reading the template and adding complementary nucleotides, creating new double helices with extraordinary accuracy. The machinery of replication has been refined over billions of years of evolution. The process in human cells is fundamentally similar to the process in bacteria, in plants, in fungi, in all forms of life that use DNA as their genetic material. The specific proteins differ in their details, but the basic mechanism remains the same. Separate the strands, use each as a template, synthesize complementary strands, proofread for errors, and repair any mistakes. This universal mechanism testifies to the common ancestry of all life on Earth. Yet despite its extraordinary accuracy, replication is not perfect. The errors that slip through, roughly 1 per cell division, are the source of new genetic variation. Most of these errors occur in non-coding DNA and have no effect. Some occur in genes but do not change the protein product because of redundancy in the genetic code. A few do change proteins, and of these, most are harmful and are eliminated by natural selection. But occasionally, very rarely, an error creates a beneficial change, and this change may spread through a population over generations. The imperfect accuracy of DNA replication, then, is not a flaw but a feature. It provides the variation upon which evolution acts, allowing life to adapt to changing environments. In the gentle darkness of the cell nucleus, the double helix unwinds, strands separate, and DNA polymerase performs its meticulous work. This process, refined over billions of years, ensures that genetic information passes faithfully from cell to cell, generation to generation, maintaining continuity while allowing for the small variations that make each individual unique. DNA contains the instructions for building proteins, but DNA itself does not directly make proteins. The information must first be copied from DNA into a related molecule called RNA. And then that RNA must be translated into the sequence of amino acids that forms a protein. This two-step process, DNA to RNA to protein, is so fundamental to biology that it is called the central dogma of molecular biology. The first step, copying DNA into RNA, is called transcription. The word is apt. Just as transcribing a document means copying it into the same language, transcription copies information from DNA's 4-letter alphabet into RNA's nearly identical 4-letter alphabet. RNA, or ribonucleic acid, is chemically similar to DNA with a few key differences. RNA contains the sugar ribose instead of deoxyribose. It uses the base uracil instead of thymine. And unlike DNA, which typically exists as a double helix, RNA is usually single-stranded. Transcription begins when a protein called RNA polymerase binds to a specific sequence of DNA called a promoter. Promoters are located just upstream of genes. They are like street signs that say, "Gene ahead." The RNA polymerase recognizes these promoter sequences and binds to the DNA at that location. Once bound, the enzyme separates the two strands of DNA, creating a small bubble of single-stranded DNA. The RNA polymerase then moves along one of the DNA strands, the template strand, reading its sequence and synthesizing a complementary strand of RNA. Where the DNA has an adenine, the RNA polymerase adds a uracil to the growing RNA strand. Where the DNA has a thymine, the enzyme adds an adenine. Where the DNA has a guanine, it adds a cytosine. And where the DNA has a cytosine, it adds a guanine. The RNA polymerase moves along the gene, unwinding the DNA ahead of it and rewinding it behind, leaving a growing strand of RNA in its wake. When the RNA polymerase reaches the end of the gene, marked by a termination signal, it releases the completed RNA molecule and detaches from the DNA. The DNA double helix reforms and the gene is ready to be transcribed again if needed. A single gene can be transcribed multiple times, producing many RNA copies, which allows the cell to make large amounts of the corresponding protein when necessary. The RNA that is transcribed from a gene is called messenger RNA, or mRNA, because it carries the genetic message from the nucleus to the ribosomes in the cytoplasm where proteins are made. But messenger RNA is not the only type of RNA in the cell. In fact, most of the RNA in a cell is not mRNA, but rather other types of RNA that play structural and catalytic roles. Ribosomal RNA, or rRNA, is a major component of ribosomes, the molecular machines that synthesize proteins. Ribosomes are made of both RNA and protein, but the ribosomal RNA is not merely structural. It actually catalyzes the chemical reaction that links amino acids together to form proteins. This discovery revolutionized our understanding of RNA, showing that it can act as an enzyme, not just as a carrier of information. Transfer RNA, or tRNA, is another crucial type of RNA. Transfer RNA molecules are small, roughly 80 nucleotides long, and fold into a characteristic cloverleaf shape. Each tRNA carries a specific amino acid and has a 3-nucleotide sequence called an anticodon that recognizes complementary sequences in mRNA. Transfer RNA molecules are the adapters that translate the language of nucleic acids, sequences of nucleotides, into the language of proteins, sequences of amino acids. In addition to these three major classes, cells contain many other types of RNA. MicroRNAs are short RNA molecules that regulate gene expression by binding to messenger RNAs and affecting their stability or translation. Small nuclear RNAs participate in processing messenger RNA. Long non-coding RNAs have diverse regulatory functions that scientists are still working to understand. RNA is not just an intermediate between DNA and protein, but a versatile molecule with many roles in the cell. The genetic code is the set of rules that translates RNA sequences into protein sequences. The code is read in units of 3 nucleotides called codons. Each codon specifies one amino acid. Since there are 4 different nucleotides in RNA, adenine, uracil, guanine, and cytosine, there are 64 possible 3-nucleotide combinations. But there are only 20 standard amino acids used to build proteins. This means the genetic code has redundancy. Multiple codons can specify the same amino acid. For example, the amino acid leucine is specified by 6 different codons: UUA, UUG, CUU, CUC, CUA, and CUG. This redundancy provides some protection against mutations. A change in the 3rd position of a codon often does not change which amino acid is incorporated into the protein. The genetic code also includes 3 stop codons, UAA, UAG, and UGA, which signal the end of translation. When the ribosome encounters a stop codon, it releases the completed protein. Remarkably, the genetic code is nearly universal. The same codons specify the same amino acids in humans, in bacteria, in plants, and fungi, in virtually all forms of life. There are a few minor variations. Mitochondria use a slightly different code, and a few unusual organisms have modified the code in small ways. But the overwhelming conservation of the genetic code across all domains of life is powerful evidence that all life on Earth shares a common ancestor. The second major step in gene expression is translation, converting the sequence of nucleotides in mRNA into a sequence of amino acids in a protein. Translation takes place at the ribosome, a large complex made of ribosomal RNA and dozens of proteins. The ribosome has 2 main sites: the A site, where new amino acids enter, and the P site, where the growing protein chain is held. Translation begins when a ribosome binds to an mRNA molecule near its beginning, at a sequence called the start codon, which is almost always AUG. A transfer RNA molecule carrying the amino acid methionine recognizes this start codon through complementary base pairing between the codon and the tRNA's anticodon. The anticodon on this particular tRNA is UAC, which pairs with the start codon AUG. Once the first tRNA is in place, the ribosome moves to the next codon on the mRNA. Another tRNA carrying the amino acid specified by this second codon enters the A site of the ribosome. Its anticodon pairs with the codon on the mRNA. Now the ribosome catalyzes the formation of a chemical bond between the first amino acid and the second amino acid. The first tRNA releases its amino acid and exits the ribosome, while the second tRNA, now carrying a chain of 2 amino acids, moves to the P site. The process repeats. The ribosome moves to the next codon, a new tRNA enters with its amino acid. A bond forms between the growing chain and the new amino acid, and the process continues. The ribosome moves along the mRNA like a molecular railroad car on a track, reading each codon in sequence and adding the corresponding amino acid to the growing protein chain. The speed is remarkable. A bacterial ribosome adds about 20 amino acids per second, while a human ribosome adds about 6 per second. When the ribosome reaches a stop codon, there is no tRNA with a matching anticodon. Instead, proteins called release factors recognize the stop codon and cause the ribosome to release the completed protein. The ribosome then separates from the mRNA, ready to begin translation again. A single mRNA molecule can be translated by many ribosomes simultaneously. As one ribosome moves along the mRNA, another can bind at the start codon and begin translating behind it. This creates structures called polysomes, strings of ribosomes all translating the same mRNA at the same time, each one producing a copy of the protein. This allows the cell to produce many copies of a protein quickly from a single mRNA molecule. The flow of information from DNA to RNA to protein is elegant and efficient. DNA remains safely tucked in the nucleus, protected from damage. When a protein is needed, RNA polymerase transcribes the corresponding gene, producing messenger RNA molecules that carry the information out to the cytoplasm. There, ribosomes translate the mRNA into proteins, using transfer RNAs as adapters to match codons with amino acids. When enough protein has been made, the cell can stop transcribing the gene, and the existing mRNA will eventually be degraded, stopping protein production. This system allows for exquisite control over gene expression. A cell can regulate how much of a protein it makes by controlling how often a gene is transcribed, how stable the mRNA is, how efficiently the mRNA is translated, and how long the protein lasts before being degraded. Different cell types express different sets of genes, which is why a neuron looks and functions differently from a muscle cell despite containing the same DNA. Development from a single fertilized egg into a complex organism requires precisely timed changes in gene expression. Certain genes must be turned on in specific cells at specific times. As you lie here in the quiet, your cells are engaged in a constant conversation between DNA, RNA, and proteins. RNA polymerases are transcribing genes, producing messenger RNAs that stream out of the nucleus into the cytoplasm. Ribosomes are translating those mRNAs, adding amino acids one by one to build the proteins your body needs. Transfer RNAs are shuttling amino acids to ribosomes, matching anticodons to codons, ensuring that the genetic information is translated accurately into molecular form. The central dogma, DNA to RNA to protein, provides the framework for understanding how genetic information is expressed. But like all good frameworks, it is not rigid. Scientists have discovered that RNA can sometimes be copied back into DNA, that proteins can influence which genes are transcribed, and that RNA can regulate other RNAs. The flow of information in cells is more complex than a simple one-way street, yet the central dogma remains fundamental. DNA stores information, RNA carries that information and performs catalytic functions, and proteins do most of the structural and enzymatic work of the cell. In the darkness of the cell, molecular machines work continuously, reading genetic information and translating it into the proteins that build and maintain your body. From the 4 letters of the genetic code, A, U, G, C, arranged in triplets, the cell constructs 20 different amino acids in precise sequences, folding them into proteins with intricate 3-dimensional structures. Each protein performs its specific function. Enzymes catalyze chemical reactions. Structural proteins provide shape and support. Transport proteins carry molecules across membranes. Regulatory proteins control gene expression. All of this emerges from the elegant translation of information, nucleotide by nucleotide, codon by codon, amino acid by amino acid. The fidelity of DNA replication and the accuracy of transcription and translation are remarkable, but they are not perfect. Errors occur, and these errors, along with other changes to the DNA sequence, are called mutations. Mutations are the ultimate source of all genetic variation, the raw material upon which evolution acts. Without mutations, all organisms would be genetically identical to their ancestors, and life would be unable to adapt to changing environments. Yet mutations can also cause disease and dysfunction. Understanding mutations requires us to examine both their molecular mechanisms and their consequences. Mutations can be classified in several ways. One classification is based on the type of change to the DNA sequence. Point mutations, also called single nucleotide variants or SNPs, involve changes to a single base pair. If an adenine-thymine base pair is changed to a guanine-cytosine pair, or vice versa, this is a point mutation. Point mutations are the most common type of mutation. There are different categories of point mutations based on their effects. A silent mutation changes a codon but does not change the amino acid that the codon specifies. This is possible because of the redundancy in the genetic code. Remember that most amino acids are specified by more than one codon. For example, if the codon UUU, which specifies the amino acid phenylalanine, mutates to UUC, the protein will still have phenylalanine at that position because UUC also codes for phenylalanine. Silent mutations have no effect on the protein's structure or function. A missense mutation changes a codon so that it specifies a different amino acid. If the mutation changes UUU to UCU, the protein will have serine instead of phenylalanine at that position. The effect of a missense mutation depends on how different the new amino acid is from the original and where in the protein the change occurs. Some missense mutations have little effect. If the new amino acid has similar chemical properties to the original, the protein may still function normally. Other missense mutations can be devastating, completely disrupting the protein's structure or function. A nonsense mutation changes a codon that specifies an amino acid into a stop codon. If translation encounters this premature stop codon, it will terminate early, producing a shortened protein that is almost always non-functional. Nonsense mutations are typically more harmful than missense mutations because they eliminate entire portions of the protein. Insertions and deletions, often called indels, involve adding or removing one or more nucleotides from the DNA sequence. If the number of nucleotides inserted or deleted is not a multiple of 3, the mutation causes a frame shift. Remember that the genetic code is read in triplets, codons. If you add or delete 1 or 2 nucleotides, all the codons downstream of the mutation are shifted, completely changing the sequence of amino acids in the protein from that point onward. Frame-shift mutations almost always destroy protein function because they alter everything after the mutation site. If the insertion or deletion involves a number of nucleotides that is a multiple of 3, it does not cause a frame shift. Instead, it simply adds or removes amino acids from the protein. These in-frame insertions or deletions can have varying effects depending on where they occur and how many amino acids are added or removed. Mutations can also be classified by their scale. The mutations we have discussed so far affect only 1 or a few nucleotides, but larger chromosomal mutations can affect long stretches of DNA or even entire chromosomes. Duplications occur when a segment of DNA is copied, creating an extra copy in the genome. Deletions can remove large segments containing multiple genes. Inversions flip a segment of DNA so that it points in the opposite direction. Translocations move a segment from one chromosome to another. These large-scale chromosomal mutations can have dramatic effects, often disrupting multiple genes simultaneously. Mutations arise through several mechanisms. Spontaneous mutations occur as a result of errors during DNA replication. Despite the accuracy of DNA polymerase and the multiple layers of proofreading and repair, mistakes occasionally slip through. Spontaneous mutations can also result from chemical changes to DNA bases. For example, cytosine can spontaneously lose an amino group, converting it to uracil. If this change is not repaired before the next round of replication, it will result in a permanent mutation. Induced mutations are caused by external factors called mutagens. Some chemicals can damage DNA, causing mutations. For instance, certain substances found in tobacco smoke can add chemical groups to DNA bases, distorting the double helix and causing errors during replication. Radiation is another potent mutagen. Ultraviolet light from the sun can cause adjacent thymine bases to bond together, creating thymine dimers that distort the DNA structure. X-rays and gamma rays can break the DNA backbone directly. Cells have repair systems to fix this damage, but if the damage is extensive or if it is not repaired correctly, mutations result. The consequences of mutations depend on where they occur and what they do. Most of the human genome is non-coding DNA, so most mutations occur in regions that do not directly encode proteins. Many of these mutations have no noticeable effect at all. Even mutations in genes often have minimal impact. Silent mutations, as we discussed, do not change the protein. Some missense mutations substitute one amino acid for another without significantly affecting protein function. But some mutations have profound effects. Sickle cell disease, for instance, is caused by a single-point mutation in the gene for hemoglobin, the protein that carries oxygen in red blood cells. This mutation changes a single amino acid. Glutamic acid is replaced by valine at position 6 of the beta-globin chain. This seemingly small change causes the hemoglobin molecules to stick together when oxygen levels are low, distorting the red blood cells into a sickle shape. These misshapen cells get stuck in small blood vessels, causing pain and damage to organs. A change of just one nucleotide out of 3 billion in the genome leads to a serious disease. Cystic fibrosis is another disease caused by mutations in a single gene. The most common mutation causing cystic fibrosis is a 3-nucleotide deletion that removes a single amino acid, phenylalanine at position 508, from a protein called CFTR. This protein normally regulates the movement of chloride ions across cell membranes. Without the phenylalanine, the protein does not fold correctly and is degraded before it can reach the cell membrane. The absence of functional CFTR leads to thick mucus in the lungs and other organs, causing the symptoms of cystic fibrosis. Not all mutations are harmful. Some have no effect, as we have discussed. Others are actually beneficial. A mutation that provides some advantage, better resistance to disease, improved metabolism, enhanced sensory abilities, may increase in frequency over generations through natural selection. The lactase persistence mutation, for example, allows some adults to digest milk sugar, a trait that became common in populations that domesticated dairy animals. The mutation provided a nutritional advantage, and so it spread through those populations. Many mutations are beneficial or harmful depending on the environment. The sickle cell mutation, despite causing disease when a person inherits 2 copies, provides protection against malaria when a person inherits only 1 copy. In regions where malaria is common, carrying 1 copy of the sickle cell mutation is advantageous because the benefit of malaria resistance outweighs the cost of having some abnormal red blood cells. In regions without malaria, the mutation is purely harmful. This is an example of balancing selection, where a mutation is maintained in a population because it is beneficial in some circumstances even though it is harmful in others. Single nucleotide polymorphisms, or SNPs, are common variations in the human genome. A SNP is a position in the genome where different individuals have different nucleotides. If 90% of people have an adenine at a particular position and 10% have a guanine, that position is a SNP. The human genome contains millions of SNPs, about 1 in every 300 to 1,000 base pairs. Most SNPs are in non-coding regions and have no effect on health or traits. But some SNPs in or near genes can affect how much protein is made or alter the protein's function. SNPs are responsible for much of the variation we see between individuals. They contribute to differences in height, hair color, eye color, and countless other traits. They also influence susceptibility to diseases. Some SNPs increase the risk of heart disease, diabetes, Alzheimer's disease, and other conditions. By studying SNPs across many individuals, scientists can identify which genetic variants are associated with particular traits or diseases, helping to unravel the genetic basis of complex characteristics. Mutations occur at a relatively constant rate over time. In humans, each person is born with about 60 to 70 new mutations that were not present in either parent. These de novo mutations arise during the production of egg and sperm cells, or in the early divisions of the embryo. Most of these new mutations have no noticeable effect. Very rarely, a new mutation causes a genetic disease in a child whose parents do not carry the mutation. Over generations, mutations accumulate. Comparing the genomes of different species allows scientists to estimate how long ago those species shared a common ancestor. The more mutations that have accumulated, the more time has passed since the species diverged. This molecular clock provides insights into evolutionary history, helping to construct the tree of life that connects all organisms. As you rest here in the darkness, mutations are occurring in cells throughout your body. Most will be in cells that die with you and will not be passed on. A few in the cells that produce eggs or sperm might be transmitted to the next generation. Most of these mutations will be harmless, making no difference to the protein products of genes or occurring in regions of the genome that do not code for proteins at all. A tiny fraction might be harmful, potentially causing genetic disease. An even tinier fraction might be beneficial, providing some advantage that natural selection might favor over many generations. Mutations are the engine of evolution, the source of the genetic diversity that allows species to adapt and change. They are also the cause of many genetic diseases, a reminder of the delicate precision required for life to function properly. In the quiet depths of the genome, changes occur, single nucleotides altered, small insertions or deletions, occasionally larger rearrangements, creating variation in an otherwise stable system, ensuring that life remains dynamic, adaptable, and ever-changing. DNA is under constant assault. Every day in every cell of your body, thousands of DNA lesions occur, damage to the chemical structure of DNA that, if left unrepaired, would lead to mutations or cell death. This damage comes from both external sources and from the normal chemical reactions taking place inside cells. Ultraviolet radiation from sunlight strikes DNA, causing chemical bonds to form between adjacent bases. Reactive oxygen molecules generated as byproducts of cellular metabolism attack DNA, breaking bonds and modifying bases. Errors during DNA replication leave mismatched base pairs or gaps in the newly synthesized strands. Yet despite this constant barrage of damage, the DNA in most cells remains remarkably stable. This stability is maintained by an elaborate network of DNA repair systems, molecular surveillance and correction mechanisms that patrol the genome, detect damage, and fix it before it becomes permanent. These repair systems are so important that defects in DNA repair genes can lead to dramatically increased rates of mutation, premature aging, and cancer. One of the most common types of spontaneous DNA damage is the loss of an amino group from cytosine, converting it to uracil. Cytosine naturally undergoes this chemical change, called deamination, at a low but steady rate. If not corrected, the uracil would pair with adenine during the next round of replication instead of with guanine, resulting in a mutation. To prevent this, cells have a repair system called base excision repair. Base excision repair begins when an enzyme called a DNA glycosylase recognizes the abnormal base, in this case, uracil. The glycosylase breaks the chemical bond connecting the uracil to the sugar phosphate backbone, removing the damaged base and leaving an empty space in the DNA strand. This empty space, called an abasic site or AP site, is recognized by another enzyme called AP endonuclease, which cuts the sugar phosphate backbone at that location. Other enzymes then remove the damaged section, and DNA polymerase fills in the gap using the opposite strand as a template. Finally, DNA ligase seals the break, restoring the DNA to its original undamaged state. Base excision repair handles many types of small-scale damage to individual bases. Cells have multiple different DNA glycosylases, each one specialized to recognize a particular type of damaged base. This allows the cell to detect and repair a wide variety of chemical modifications to DNA. More extensive damage requires a different repair system called nucleotide excision repair. This system handles bulky lesions that distort the DNA double helix. One common type of damage repaired by this pathway is thymine dimers, which form when ultraviolet light causes two adjacent thymine bases to bond together. These dimers create a kink in the DNA that blocks replication and transcription. Nucleotide excision repair is more complex than base excision repair. First, a protein complex scans the DNA looking for distortions in the double helix. When it finds damage, it binds to the site. Enzymes then cut the damaged strand on both sides of the lesion, removing a segment of about 24 to 32 nucleotides that includes the damage. DNA polymerase fills in the gap using the undamaged complementary strand as a template, and DNA ligase seals the final break. Nucleotide excision repair is particularly important for protecting against sun damage. People with defects in nucleotide excision repair genes suffer from a condition called xeroderma pigmentosum. Their cells cannot repair thymine dimers and other UV-induced damage, leading to an extremely high risk of skin cancer, sometimes hundreds of times higher than in the general population. This tragic disease demonstrates the crucial importance of DNA repair in maintaining genome stability. We have already discussed mismatch repair in the context of DNA replication. This system corrects base pair mismatches that escape the proofreading activity of DNA polymerase. Mismatch repair proteins scan newly replicated DNA, looking for places where the bases are not properly paired. When a mismatch is found, the system must determine which strand contains the error. The repair machinery then removes a segment of the new strand containing the mismatch and resynthesizes it correctly. Defects in mismatch repair lead to a dramatic increase in mutation rate. People who inherit defective mismatch repair genes develop a condition called Lynch syndrome, which causes a very high risk of colon cancer and other cancers. The tumor cells in these patients accumulate mutations much faster than normal, leading to rapid cancer development. Perhaps the most dangerous type of DNA damage is a double-strand break, a complete break in both strands of the DNA double helix. Double-strand breaks can be caused by ionizing radiation, certain chemicals, or errors during DNA replication. These breaks are particularly hazardous because they can lead to chromosome rearrangements or loss of genetic information. Cells have two main strategies for repairing double-strand breaks. The first, called homologous recombination, uses the undamaged sister chromosome or sister chromatid as a template to accurately repair the break. This mechanism is most active during and shortly after DNA replication, when the sister chromatid is nearby and available to serve as a template. Homologous recombination is remarkably accurate, restoring the original DNA sequence. The second strategy, called non-homologous end joining, simply connects the broken ends back together without using a template. This is faster than homologous recombination but less accurate. Small insertions or deletions often occur at the repair site. Non-homologous end joining is the main repair pathway in cells that are not actively replicating their DNA. Mutations in genes involved in repairing double-strand breaks can have severe consequences. The BRCA1 and BRCA2 genes, for instance, play important roles in homologous recombination. Women who inherit mutations in these genes have a greatly elevated risk of breast and ovarian cancer because their cells cannot accurately repair double-strand breaks. The accumulation of unrepaired or incorrectly repaired breaks leads to chromosome instability and eventually to cancer. DNA repair systems must work constantly to keep up with the ongoing damage. Estimates suggest that each cell in your body experiences tens of thousands of DNA lesions per day. The vast majority of these are repaired correctly, allowing the cell to continue functioning normally. But occasionally, damage occurs in a region of the genome that is being replicated before repair can take place, fixing the damage as a permanent mutation. Over a lifetime, mutations accumulate in cells throughout the body. The efficiency of DNA repair declines with age. Older cells are less capable of detecting and correcting damage than younger cells. This decline in repair capacity contributes to the increased cancer risk that comes with aging. It may also contribute to other aspects of aging, as accumulated DNA damage affects gene expression and cellular function. Cancer cells often have defects in DNA repair. These defects allow mutations to accumulate rapidly, driving the evolution of the cancer. Ironically, these same repair defects can make cancer cells vulnerable to certain treatments. For example, cancer cells with defects in homologous recombination are particularly sensitive to drugs that cause additional DNA damage because they cannot repair that damage effectively. This principle, exploiting the repair defects in cancer cells, is the basis for some targeted cancer therapies. DNA repair systems are evolutionarily ancient. Even bacteria have multiple repair pathways to protect their genomes. The basic mechanisms of base excision repair, nucleotide excision repair, and mismatch repair are similar across all domains of life. This conservation reflects the fundamental importance of maintaining genome stability. Without effective DNA repair, mutation rates would be so high that genetic information could not be transmitted faithfully from one generation to the next. As you rest in the quiet darkness, repair proteins are moving through the DNA in your cells, scanning for damage. DNA glycosylases are plucking out modified bases. Nucleotide excision repair complexes are recognizing and removing bulky lesions. Mismatch repair systems are correcting replication errors. Double-strand break repair pathways are mending broken chromosomes. This invisible molecular maintenance work proceeds continuously, protecting the integrity of your genome. The balance between DNA damage and DNA repair shapes the fate of every cell. When damage overwhelms repair capacity, mutations accumulate, potentially leading to cancer or cell death. When repair systems function properly, cells maintain genetic stability across countless divisions. The surveillance and correction mechanisms that patrol your DNA represent billions of years of evolutionary refinement, a molecular quality control system of extraordinary sophistication. In the nucleus of each cell, damage and repair are locked in constant equilibrium. Bases are damaged and excised, lesions distort the helix and are cut away. Breaks occur and are rejoined. The genome is not a static, unchanging text, but a dynamic structure constantly being maintained and repaired. The stability we perceive, the faithful transmission of genetic information from cell to cell and generation to generation, emerges from this ongoing process of detection and correction, damage and repair, ensuring that the instructions for life remain readable despite the chaos of chemistry. The DNA sequence in your genome is essentially the same in every cell of your body. A neuron in your brain, a muscle cell in your heart, and a white blood cell in your bloodstream all contain the same 3 billion base pairs arranged in the same order. Yet these cells look different, behave differently, and perform entirely different functions. This paradox, identical genomes producing different cell types, is resolved by epigenetics, the study of changes in gene expression that do not involve changes to the DNA sequence itself. The term epigenetics means above genetics or beyond genetics. Epigenetic modifications are chemical changes to DNA or to the histone proteins around which DNA wraps. These modifications do not alter the sequence of A's, T's, G's, and C's, but they profoundly affect which genes are turned on or off. Through epigenetic mechanisms, cells can activate some genes and silence others, creating the diversity of cell types needed to build and maintain a complex organism. The most studied epigenetic modification is DNA methylation. This involves adding a small chemical group called a methyl group, one carbon atom bonded to 3 hydrogen atoms, to certain DNA bases. In mammals, methylation occurs almost exclusively at cytosine bases that are followed by guanine bases, a pattern abbreviated as CpG. These CpG sites are not evenly distributed throughout the genome, but tend to cluster in regions called CpG islands, which are often found near the beginnings of genes. DNA methylation is carried out by enzymes called DNA methyltransferases. These enzymes transfer methyl groups from a donor molecule to cytosine bases in DNA. Once a cytosine is methylated, it remains methylated through subsequent rounds of DNA replication because maintenance methyltransferases recognize methylated CpG sites on the template strand and add methyl groups to the corresponding sites on the newly synthesized strand. This allows methylation patterns to be inherited when cells divide. Methylated DNA is generally associated with gene silencing. When the CpG island near a gene's promoter is heavily methylated, that gene is usually turned off. The methyl groups do not directly prevent transcription, but they recruit proteins that do. These proteins bind to methylated DNA and create a tightly packed chromatin structure that is inaccessible to RNA polymerase and the other machinery needed for transcription. The gene is effectively locked away, unable to be expressed. DNA methylation plays crucial roles in normal development and cell differentiation. As an embryo develops, different cells activate different sets of genes by demethylating some regions and maintaining methylation in others. Once a cell commits to becoming a particular type, a neuron for instance, it establishes a pattern of methylation that keeps inappropriate genes silenced. The genes for muscle proteins remain methylated and inactive in neurons, while the genes for neurotransmitter receptors are demethylated and active. This epigenetic programming allows cells with identical genomes to maintain distinct identities. DNA methylation is also essential for a process called genomic imprinting. For most genes, the copy inherited from your mother and the copy from your father are both active. But for a small number of genes, perhaps 100 to 200 in humans, only one copy is expressed. Which copy is active depends on which parent it came from. Imprinted genes have methylation patterns that differ between the maternal and paternal chromosomes, silencing one copy while allowing the other to be expressed. Errors in imprinting can cause developmental disorders, demonstrating the importance of proper epigenetic regulation. Histone modifications represent another major category of epigenetic regulation. Recall that DNA wraps around histone proteins to form nucleosomes. The histone proteins have tails, stretches of amino acids that protrude from the nucleosome core. These tails can be modified by adding various chemical groups, methyl groups, acetyl groups, phosphate groups, and others. Each type of modification has different effects on chromatin structure and gene expression. Histone acetylation generally promotes gene expression. Acetyl groups are added to lysine amino acids in histone tails by enzymes called histone acetyltransferases. These acetyl groups neutralize the positive charge on lysine, weakening the interaction between histones and DNA. This loosens the chromatin structure, making DNA more accessible to transcription factors and RNA polymerase. Conversely, removing acetyl groups, a process carried out by histone deacetylases, tightens chromatin and generally reduces gene expression. Histone methylation is more complex because its effects depend on which lysine is modified and how many methyl groups are added. Some histone methylation marks are associated with active genes, while others are associated with silenced genes. For example, trimethylation of histone H3 at lysine 4 is typically found near active genes, while trimethylation of histone H3 at lysine 9 is associated with silent heterochromatic regions. The combination of different histone modifications creates a complex code that influences whether a gene is expressed or silenced. Chromatin remodeling complexes can also change gene expression without altering DNA sequence. These protein machines use energy to slide, eject, or restructure nucleosomes, changing how tightly DNA is packaged. By repositioning nucleosomes, these complexes can expose or hide regulatory sequences that control gene expression. The interplay between DNA methylation, histone modifications, and chromatin remodeling creates a sophisticated regulatory system that controls which genes are active in each cell type. Remarkably, epigenetic modifications can be influenced by environmental factors. Diet, stress, toxins, and other environmental exposures can alter DNA methylation patterns and histone modifications. For example, during a Dutch famine in World War II, pregnant women who experienced severe malnutrition had children with altered methylation patterns in certain genes. These epigenetic changes persisted into adulthood and were associated with increased risk of metabolic diseases. The children's experiences in the womb, specifically their mother's nutritional status, had left a lasting molecular mark on their genomes. This environmental responsiveness of the epigenome has profound implications. It means that experiences can change how genes are expressed, potentially affecting health and behavior long after the experience has passed. Identical twins who share the same DNA sequence accumulate different epigenetic modifications over their lifetimes as they have different experiences. By old age, the epigenomes of identical twins can be quite different, which may explain why their health outcomes sometimes diverge despite their genetic similarity. Some epigenetic modifications can even be transmitted from parent to offspring. During the formation of eggs and sperm, most epigenetic marks are erased and then reset in the next generation. However, some marks escape this erasure and are passed on. This means that in certain cases, the experiences of parents or even grandparents can affect the gene expression patterns in their descendants. This transgenerational epigenetic inheritance is well documented in plants and worms, and evidence is accumulating that it also occurs in mammals, including humans. Epigenetic changes play important roles in aging. As cells divide over a lifetime, their epigenetic patterns gradually change. Some genes that should be active become methylated and silenced, while some genes that should be silent become demethylated and active. This epigenetic drift may contribute to the functional decline of tissues with age. Scientists are investigating whether reversing age-related epigenetic changes could slow or even reverse aspects of aging. Cancer is fundamentally a disease of altered gene expression, and epigenetic changes are a major contributor. In many cancers, genes that normally suppress tumor growth become hypermethylated and silenced, while genes that promote cell division become demethylated and overactive. These epigenetic changes can be as important as DNA mutations in driving cancer development. Unlike mutations, which are permanent changes to DNA sequence, epigenetic modifications are potentially reversible. This has led to the development of epigenetic drugs that target DNA methyltransferases or histone-modifying enzymes. reactivating silenced genes or quieting overactive ones. The study of epigenetics has transformed our understanding of heredity. Inheritance is not simply a matter of passing on DNA sequences. The epigenetic modifications on that DNA, the patterns of methylation, the histone modifications, the three-dimensional organization of chromatin, are also transmitted from cell to cell and sometimes from parent to offspring. These modifications carry information about which genes should be active and which should be silent, information that is crucial for proper development and function. As you lie here in the darkness, epigenetic modifications are being added and removed throughout your genome. Methyl groups are being attached to cytosines in regions that should be silenced. Acetyl groups are being added to histone tails near genes that need to be expressed. Chromatin remodeling complexes are repositioning nucleosomes, opening up some regions of DNA and closing others. These changes are responding to signals from within your cells and from your environment, the food you ate today, your stress levels, your sleep quality, your physical activity. Your epigenome is not fixed but fluid, adjusting constantly to your circumstances. It is a bridge between your unchanging genome and your ever-changing environment, allowing your cells to respond to experiences and conditions without altering the fundamental DNA sequence. In the quiet chemistry of the nucleus, methyl groups and acetyl groups and phosphate groups are added and removed. Chromatin loosens and tightens. Genes wake and sleep. This dynamic regulation, layered on top of the static DNA sequence, transforms a fixed text into a responsive document, allowing the same genome to generate different cell types, respond to different environments, and adapt to different stages of life. At the ends of each chromosome are specialized structures called telomeres. These sequences serve as protective caps, much like the plastic tips on shoelaces that prevent the laces from fraying. Telomeres do not contain genes. Instead, they consist of the same short DNA sequence repeated thousands of times. In humans, this sequence is T-tag, and it repeats approximately 2,500 times at the end of a newly formed chromosome, adding up to about 15,000 base pairs of telomeric DNA. The discovery of telomeres and their function represents one of the most important insights into cellular aging. The story begins with a problem inherent in how DNA replication works. Remember that DNA polymerase can only add nucleotides to the 3' end of a growing strand and requires a primer to begin synthesis. At the very end of a linear chromosome, when the RNA primer for the last Okazaki fragment is removed, there is no way to fill in that gap. The DNA polymerase has no primer upstream of the gap to extend. This means that with each round of replication, the chromosome becomes slightly shorter at its ends. This progressive shortening is called the end replication problem. If it went unchecked, chromosomes would lose essential genetic information with each cell division, eventually leading to malfunction and death. Telomeres solve this problem by providing a buffer of non-coding DNA that can be lost without losing genes. The telomeric repeats absorb the shortening, protecting the coding regions of the chromosome, but telomeres themselves still shorten with each cell division. In human cells that lack a special enzyme called telomerase, telomeres lose approximately 50 to 200 base pairs per cell division. After many divisions, telomeres become critically short. When this happens, the cell recognizes the shortened telomeres as damaged DNA and stops dividing, entering a state called senescence. Senescent cells remain alive but can no longer divide. Eventually, if telomeres become short enough, the cell may undergo programmed death. This progressive telomere shortening acts as a molecular clock, counting down the number of times a cell can divide. Most human cells can divide approximately 50 to 70 times before their telomeres become too short, a limit called the Hayflick limit after the scientist Leonard Hayflick who discovered it. This built-in division counter is thought to be a protective mechanism against cancer. Cancer requires cells to divide uncontrollably, and the telomere clock limits how many times a cell can divide before it must stop. A normal cell that accumulates cancer-causing mutations will eventually reach its division limit and stop proliferating before it can form a tumor. However, some cells can maintain their telomeres and divide indefinitely. These include stem cells, which must be able to divide throughout an organism's lifetime to replenish tissues. Germ cells, the cells that produce eggs and sperm, also maintain their telomeres because telomere length must be reset in each generation. These special cells express telomerase, an enzyme that adds telomeric repeats to chromosome ends, counteracting the shortening that occurs during replication. Telomerase is a remarkable enzyme, part protein and part RNA. The RNA component carries a template sequence that is complementary to the telomeric repeat. Telomerase binds to the end of a chromosome, uses its RNA template to add new copies of the telomeric sequence, and then moves along to extend the telomere further. By repeatedly adding 6 base pair repeats, telomerase can elongate telomeres, allowing cells to continue dividing without reaching the Hayflick limit. In most adult human tissues, telomerase is not active, and telomeres shorten with each cell division. This telomere shortening is associated with aging. Older individuals generally have shorter telomeres than younger individuals. People with unusually short telomeres for their age are at increased risk for age-related diseases, including cardiovascular disease, diabetes, and certain cancers. Whether short telomeres cause these diseases or are simply a marker of cellular damage from other sources remains an area of active research. Several factors influence the rate of telomere shortening. Chronic stress is associated with faster telomere loss. Studies have found that people experiencing prolonged psychological stress, such as those caring for chronically ill family members, have shorter telomeres than age-matched controls. The mechanism appears to involve oxidative stress and inflammation, both of which can damage telomeric DNA. Exercise and healthy lifestyle habits are associated with longer telomeres. Smoking, obesity, and poor diet are associated with shorter telomeres. These correlations suggest that telomere length reflects, at least in part, the cumulative effects of lifestyle and environmental factors on cellular health. Cancer cells face a particular challenge related to telomeres. To grow into a large tumor, cancer cells must divide far more than the 50 to 70 times permitted by normal telomere shortening. Most cancer cells solve this problem by reactivating telomerase. In approximately 85 to 95% of human cancers, telomerase is turned back on, allowing the cancer cells to divide indefinitely. The remaining cancers use a different mechanism called alternative lengthening of telomeres, or ALT, which maintains telomeres through recombination rather than telomerase. The fact that most cancers depend on telomerase activity makes this enzyme an attractive target for cancer therapy. Drugs that inhibit telomerase could potentially stop cancer cells from maintaining their telomeres, causing them to eventually stop dividing. However, such drugs would also affect the stem cells and other normal cells that require telomerase, so developing telomerase inhibitors that specifically target cancer cells remains challenging. Some rare genetic diseases are caused by mutations in telomerase genes or in other genes involved in telomere maintenance. Dyskeratosis congenita is one such disorder. People with this condition have mutations that reduce telomerase activity, leading to very short telomeres even in childhood. This causes premature aging of certain tissues, particularly those that must divide frequently, such as bone marrow and the lining of the digestive tract. Patients develop anemia, immune deficiencies, and an increased risk of cancer, and many do not survive past middle age. These telomere disorders provide tragic but clear evidence that telomere maintenance is essential for normal tissue function. Researchers have investigated whether extending telomeres could slow aging or treat age-related diseases. In laboratory mice, activating telomerase in adult tissues has shown some benefits, including improved function of certain organs. However, these mice also developed more cancers, consistent with the idea that telomere shortening protects against cancer by limiting cell division. The relationship between telomeres and aging is complex. Telomeres are not simply a cause of aging, but rather one component of a multifaceted process. Telomere length in humans varies considerably between individuals, even at birth. Newborns whose mothers experienced high levels of stress during pregnancy tend to have shorter telomeres. Telomere length is also partially heritable. Longer telomeres in parents are associated with longer telomeres in their children. This variation in starting telomere length may influence lifespan and health, though the magnitude of this effect is still being determined. As you rest here in the quiet, your telomeres are silently marking the passage of cellular time. In tissues where cells divide frequently, the lining of your intestines, your bone marrow, your skin, telomeres are shortening with each division. In your stem cells, telomerase is active, maintaining telomeres so these essential cells can continue replenishing your tissues. In most of your cells, which divide rarely or not at all, telomeres remain stable, having shortened during earlier periods of development and growth. The telomeres at the ends of your chromosomes are molecular historians, recording in their length the division history of each cell. They are countdown timers measuring out the remaining divisions available to each cell line. They are protective buffers, sacrificing themselves to preserve the genes they surround. And they are constraints on cancer, preventing cells from dividing indefinitely unless they find a way to maintain their telomeres. In the darkness of the nucleus, at the very tips of each chromosome, telomeric repeats extend into space. Tag, 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 thousands of times, a repetitive sequence that serves no coding function but performs an essential protective role. With each cell division in tissues without telomerase, these repeats become fewer, the buffer zone shrinks, and the countdown continues. This molecular clock, ticking away in the ends of chromosomes, links the division capacity of cells to the aging of tissues, connecting microscopic events in DNA replication to the visible passage of time in living organisms. Within each of your cells outside the nucleus lies a different genome, one that is smaller, circular, and inherited only from your mother. This is the mitochondrial genome. Contained within the mitochondria, the organelles that generate most of the energy your cells need to function. Mitochondria are often called the powerhouses of the cell, and their DNA tells an ancient story of symbiosis, inheritance, and human migration. Mitochondria are believed to have originated more than 1 billion years ago when a primitive cell engulfed a bacterium capable of using oxygen to produce energy. Rather than being digested, this bacterium established a symbiotic relationship with its host, providing energy in exchange for a protected environment. Over millions of years, most of the bacterial genes were transferred to the host cell's nucleus, but a small genome remained in what became the mitochondrion. This mitochondrial genome still retains some bacterial characteristics, including its circular shape and the absence of the histone proteins found in nuclear DNA. The human mitochondrial genome is remarkably compact. It contains only about 16,500 base pairs, more than 100,000 times smaller than the nuclear genome. Despite this small size, mitochondrial DNA encodes 37 genes. 13 of these genes code for proteins that are essential components of the electron transport chain, the system that generates ATP, the cell's energy currency. The other genes encode transfer RNAs and ribosomal RNAs needed for protein synthesis within the mitochondria. The mitochondrial genome is so compact that genes are packed tightly together with little space between them, and some genes even overlap slightly. Each mitochondrion contains multiple copies of its genome, usually 2 to 10. Each cell contains many mitochondria, anywhere from a few dozen to several thousand, depending on the cell's energy needs. Cells with high energy requirements, such as muscle cells and neurons, have more mitochondria than cells with lower energy needs. This means that each cell contains hundreds to thousands of copies of the mitochondrial genome, far more than the 2 copies of each nuclear gene. The inheritance of mitochondrial DNA is fundamentally different from the inheritance of nuclear DNA. Nuclear DNA comes from both parents, half from your mother via the egg and half from your father via the sperm. But mitochondrial DNA comes almost exclusively from your mother. The egg cell is large and contains thousands of mitochondria, each carrying multiple copies of the mitochondrial genome. The sperm cell also contains mitochondria located in its tail to provide energy for swimming. But these paternal mitochondria are typically destroyed after fertilization. The mechanism of this destruction has been studied extensively. After a sperm fertilizes an egg, the paternal mitochondria are marked with a protein tag that signals them for degradation. The egg cell's machinery recognizes this tag and destroys the paternal mitochondria, ensuring that only maternal mitochondria remain. This process is so efficient that paternal mitochondrial DNA is rarely, if ever, passed on. There are rare exceptions to strict maternal inheritance. In 2018, researchers reported a few families in which mitochondrial DNA appeared to be inherited from both parents. This biparental inheritance is extremely uncommon and appears to require specific mutations that disrupt the normal machinery that destroys paternal mitochondria. For the vast majority of people, mitochondrial DNA is inherited solely from the mother. This maternal inheritance pattern has profound implications for human genetics and evolutionary biology. Because mitochondrial DNA passes from mother to children without recombining with paternal DNA, it accumulates mutations over generations in a predictable way. Scientists can use these mutations as a molecular clock, comparing mitochondrial DNA sequences from different individuals to estimate how long ago they shared a common maternal ancestor. These studies have traced all human mitochondrial DNA back to a woman who lived in Africa approximately 150,000 to 200,000 years ago. This woman, often called mitochondrial Eve, was not the only woman alive at that time, but she is the only woman from that era whose mitochondrial lineage has survived in modern humans. All other mitochondrial lineages from that time have gone extinct. Every person alive today carries mitochondrial DNA descended from hers. Mitochondrial DNA has been invaluable for tracing human migration patterns. As humans spread out of Africa and populated the rest of the world, different populations accumulated different mutations in their mitochondrial genomes. By analyzing these mutations, scientists can track the routes ancient humans took as they migrated across continents. Mitochondrial DNA studies have revealed multiple waves of human migration out of Africa, the peopling of Europe and Asia, the settlement of the Americas, and the colonization of remote Pacific islands. Mutations in mitochondrial DNA can cause serious diseases. Because mitochondria generate most of the cell's energy, mutations that impair this function particularly affect tissues with high energy demands. Mitochondrial diseases often involve muscle weakness, neurological problems, vision loss, hearing loss, and heart problems. These diseases show unusual inheritance patterns because of the maternal transmission of mitochondrial DNA. Affected mothers pass the disease to all their children, but affected fathers do not pass it on at all. The severity of mitochondrial diseases can vary considerably, even within the same family. This variability arises from heteroplasmy, the presence of both normal and mutant mitochondrial DNA within a single cell or individual. Because each cell contains hundreds or thousands of copies of the mitochondrial genome, a mutation might affect only some of those copies. A cell with 10% mutant mitochondrial DNA and 90% normal might function normally, while a cell with 80% mutant DNA might be severely impaired. The proportion of mutant to normal mitochondrial DNA can shift over time and can vary between tissues. When a cell divides, the mitochondria are distributed randomly to the daughter cells, so the proportion of mutant DNA can increase or decrease through chance. This random distribution means that even children of the same mother can inherit different proportions of mutant mitochondrial DNA, leading to different disease severity. The mutation rate in mitochondrial DNA is higher than in nuclear DNA. Mitochondria generate reactive oxygen molecules as byproducts of energy production, and these molecules can damage DNA. Mitochondrial DNA is located near where these reactive molecules are produced, exposing it to high levels of oxidative damage. Additionally, the DNA repair systems in mitochondria are less sophisticated than those in the nucleus. These factors combine to create a mutation rate in mitochondrial DNA that is approximately 10 to 20 times higher than in nuclear DNA. Some scientists believe that accumulation of mutations in mitochondrial DNA contributes to aging. As mutations build up over a lifetime, mitochondrial function may decline, reducing the cell's ability to produce energy efficiently. This mitochondrial decline could contribute to age-related loss of muscle strength, neurological function, and overall vitality. However, the extent to which mitochondrial mutations cause aging versus simply being a consequence of aging remains debated. Recent advances in reproductive technology have made it possible to prevent transmission of mitochondrial diseases through a procedure called mitochondrial replacement therapy. In this technique, the nucleus from an egg with mutant mitochondria is transferred to a donor egg that has had its nucleus removed but retains healthy mitochondria. The resulting egg has nuclear DNA from the intended mother but mitochondrial DNA from the donor. When this egg is fertilized and develops, the child will have nuclear DNA from both parents but mitochondrial DNA from the donor, avoiding the mitochondrial disease. This technique, sometimes called three-parent IVF, raises ethical questions but offers hope for families affected by severe mitochondrial diseases. Mitochondrial DNA has also proven valuable in forensics and archaeology, because each cell contains many copies of mitochondrial DNA compared to only 2 copies of nuclear DNA. Mitochondrial DNA is more likely to be recovered from degraded samples such as old bones or crime scene evidence. The maternal inheritance pattern means that mitochondrial DNA from an unknown sample can be compared to maternal relatives to establish identity. This technique has been used to identify remains of historical figures and victims of mass disasters. As you rest here in the darkness, your mitochondria are working continuously, using the instructions in their small circular genomes to produce the protein complexes that generate ATP. These organelles, descendants of ancient bacteria, retain their own genetic material separate from the DNA in your nucleus. The mitochondrial DNA you carry came from your mother, and hers from her mother, in an unbroken maternal line stretching back thousands of generations. In each mitochondrion, the circular genome floats in the matrix, unprotected by histones, slowly accumulating mutations at a rate faster than nuclear DNA. These organelles divide and fuse, their genomes replicate independently of nuclear DNA. And when cells divide, they are distributed to daughter cells in a way that can shift the balance between normal and mutant genomes. Your energy, your cellular vitality, depends on the proper function of these ancient bacterial genomes that became permanent residents in your cells more than 1 billion years ago. The story of mitochondrial DNA is a story of ancient symbiosis, of maternal inheritance, of human migration across continents, and of the constant generation of energy that powers every cellular process in your body. It is a reminder that you carry within you not one genome but two. One nuclear, inherited from both parents, containing most of your genes, and one mitochondrial, inherited only from your mother, small but essential, linking you to an unbroken maternal chain that extends back to the origins of humanity and beyond. Closing. We have traveled through the fundamental landscape of genetics and DNA, from the discovery of the double helix to the protective caps of telomeres, from the central dogma of molecular biology to the maternal legacy of mitochondrial DNA. We have explored how DNA is packaged and organized, how it replicates with extraordinary precision, how it is read and translated into proteins, how it changes through mutation, how it is repaired when damaged, and how it is regulated through epigenetic modifications. The genome is not simply a static instruction manual, but a dynamic, responsive document. It is damaged and repaired constantly. It is read and silenced through epigenetic marks that respond to environment and experience. Its telomeres shorten with each cell division, marking cellular time. Its mitochondrial component, inherited solely from mothers, links each person to deep ancestral lineages. Understanding DNA and genetics helps us understand ourselves, where we came from, how we develop, why we age, and how we are connected to all other living things. The 4-letter alphabet of DNA, arranged in 3 billion base pairs, contains the instructions that made you, that maintain you, and that you will pass on to the next generation. As you drift towards sleep, let these thoughts settle gently. In the quiet nucleus of each cell, the double helix rests, wound around histones folded into chromosomes, silently storing the information of life. If you found this journey through the science of genetics calming and informative, consider subscribing to Nighttime Science for more gentle explorations of the natural world.